Hello, my name is Joshua Mugabe. I welcome you to Linkage and Crossing Over. Why is not Mendel's second law always valid for two or more phenotypical traits of an individual? Mendel's second law, the law of independent assortment, is valid for genes located on different chromosomes. These genes, during meiosis, they segregate independently. Mendel's second law, however, is not valid for phenotypical features conditioned by genes located on the same chromosome. Genes under linkage. Since these genes are known as linked genes, they do not separate in meiosis, except for the phenomenon of crossing over. Why is Drosophila a convenient animal for the study of linked genes? The fruit fly Drosophila is suitable for the study of genetics because it presents many distinct traits, but only 4 chromosomes. It has 1 sex chromosome and 3 autosomes. What is linkage? Two genes are said to be under linkage or linked when they reside in the same chromosome. For example, the research of human genome discovered that factor 3 of clotting gene and factor 5 of clotting genes are located on the same chromosome. That is human chromosome 1. Whereas factor 7 gene is not linked to these genes since it is located on chromosome 13. What is crossing over? How is meiosis related to this phenomenon? Linked alleles, for example, capital A, small b, and small a, capital B, form the gametes AB and small ab that maintain the linkage of the alleles. This type of linkage is called complete linkage. In the first division of meiosis, meiosis 1, however, crossing over phenomenon may occur. Chromosomes from a pair of homologous may exchange extremities, and some once linked alleles, for example, AB and AB, recombine to form different gametes. In the case, capital A, capital B, and small a, small b. Crossing over may happen when the arms of the chromatids of each homologous are paired during meiosis. Matching portion of the extremities of the two non-sister chromatids, one from one homologous of the pair, they break and the pieces are exchanged, each of them becoming part of the arm of the other chromatid. For example, allele A is situated in a side of the arm relating to the point of breaking, and allele B is located in the other side. They will be separated and the gametes capital A, capital B, small a, small b will be formed instead of capital A, small b, and small a, capital B. The percentage of recombinant gametes relating to the normal gametes depends upon the crossing over rate, that in its turn depends upon how far distant the given alleles are in the chromosome. In genetic recombination by crossing over, what is the difference between the parental gametes and recombinant gametes? Parental gametes are those gametes that maintain the original linkage of genes, the alleles, in the chromosome. Recombinant gametes are those in which the original linkage is undone due to exchange of the chromosomal pieces by crossing over during meiosis. What is recombination frequency? Recombination frequency or crossing over rate is the percentage of recombinant gametes made by crossing over in relation to the number of parental gametes made. It is always referred to two genes located in the same chromosome. Why does recombination frequency of genes vary with distance between them in the chromosome? The further the distance between the loci of the two genes in the chromosome, the higher the recombination frequency between these genes. This is true because once alleles are nearer in the chromosome, it is more probable that they are kept united when chromosomal extremities are exchanged by crossing over. On the other hand, if they are further apart, it will be easier for them to separate by crossing over. What is a centimorgan? Centimorgan, or recombination unit, by convention is a distance between the two linked genes that corresponds to 1% of recombination frequency of these genes. How can the concept of recombination frequency be used in genetic mapping? Genetic mapping is the determination of the location of the genes in a chromosome. By determining the recombination frequency between several different linked genes, it is possible to estimate the distance between them in the chromosome. For example, gene A has a recombination frequency of 20% with gene B. This gene B has a recombination frequency of 5% with gene C. This gene C has a recombination frequency of 15% with gene A. It is possible to assert that gene A is 20 centimorgan distant from gene B, and that between them lies the gene C at 15 centimorgan of the distance from the gene A. Is crossing over important for the diversity of biological evolution? Sexual reproduction and recombination of linked genes, what we call crossing over, are, along with the mutation, are the main instrument of biological variability. Sexual reproduction allows the combination between genes situated in different chromosomes. Crossing over, however, is the only means to provide recombination of alleles located on the same chromosomes. Crossing over probably emerged and has been maintained by the evolution because of its importance to the biological diversity. Thank you very much for participating in linkage and crossing over. Please do like, share, and subscribe.